Thanks, Kevin, and good morning to everyone here. And we're about to embark on a fairly sort of busy roadshow with the aim of raising a lot of money to fund programs that could potentially cure blindness. So it's an important mission that we're on. Let me tell you a little bit about the company. We're a company that has been in the process of the last 6 months of relaunching, and the company was one of the pioneers in cell therapy technology in the United States, and had been involved in a number of programs over the years, including disease states like multiple sclerosis and lupus nephritis, where cell therapy can potentially play a role. But when I joined the company, what excited me was they had really good clinical data for treating macular degeneration with a cell therapy transplant. One thing I would just like to mention, and I'm going to keep coming back to this during the presentation, is we are not a stem cell company. We use stem cells as a raw material to manufacture fully differentiated cells, and what goes into the back of the eye in this case is what's known as retinal pigmented epithelium. And our technology is being able to manufacture the final cells that you need to work in the back of the eye from a stem cell raw material. And we have some really good opportunities because ophthalmology is a huge market right now. There are I'm told there's two hot areas in the pharmaceutical world right now. One is orphan drug diseases, and the other one is ophthalmic products. And in actual fact, our Stargardt's macular degeneration product here is an orphan indication for this therapy, and it's the program that's going to be most advanced. But behind that, there comes a category which is dry AMD, and we'll talk about this in a few minutes as well. So we're a company with the lead orphan indication, but potentially a curative therapy for dry AMD, as you'll hear later. There's a third disease category that we're tackling, which is a disease I'd actually only heard of when I came to this company, which is called myopic macular degeneration, and it's basically people with very short sight develop macular degeneration as a result of that, and it's prevalent particularly in China and Japan, where it's the second cause of blindness. So we're developing products for potentially big markets. Now, I'd like to think that at some point in the future this company could be like Regeneron. I certainly appreciate that in terms of the market cap. But right now we're a $230 million market cap, looking to raise financing in order to be able to progress these programs. So the reason that we are here today is, and the reason I joined the company, as Kevin mentioned, I had worked for another company that had finance with Jefferies, but I saw data when I was interviewing, which was just published in The Lancet. Now, for those of you that are not familiar with the medical literature, The Lancet is probably the oldest medical journal in the world. It's published out of London. It's the leading journal, in my opinion, in terms of medical reporting. And what we did was have a publication in there on October the 15th of this year. And we showed for the first time in any disease indication that we could use cell therapy transplants. And the second thing was we were able to show evidence of efficacy with this therapy. As you'll see later on, patients did start to improve their vision once they'd been transplanted, which is a huge benefit. And Eddie Anglade will get up later on and talk about the medical side of it. For those of you who are reading this slide carefully, you'll see that we have Steve Schwartz quoted here. Steve is our primary investigator on our clinical trials, and he was the lead author in the publication in The Lancet. Steve is also one of the founders of Avalanche Biotechnologies, which has been a big success. Another ophthalmic play. It's about an $800 million market cap. And this is what we're doing. We're addressing macular degeneration with groundbreaking what we call RPE therapy. That's retinal pigmented epithelium. We're just about to kick off the phase 2 studies. So the phase 2 studies are starting in Stargardt's in actually just a few weeks' time, before the end of this year. And that study is going to involve about 100 patients. It's potentially a registration study, certainly in Europe, because it's an orphan indication. And over in Europe, we believe we might be able to get that product to the market towards the end of 2018, but we're targeting 2019. The dry AMD program starts its phase 2 in Q2 of next year, and that's a big market opportunity. Eddie will take you through the details, but those are the two most important studies, and actually is probably the bulk of the use of proceeds of this offering. We also, as we were one of the founders of cell therapy technology, the work that started at the company is based on the work of a guy called Dr. Robert Lanza, and Bob still works at the company as our Chief Scientific Officer. He was nominated in Time magazine this year as one of the most 100 influential people in the country, and that was for his scientific endeavours on cell therapy. So Bob started this work. What we're now doing is translating that discovery into therapies through the development program. We have stem cells as the starting material. This is what I mentioned earlier, and this is an important point to keep hammering home to people. The fact is that the fully differentiated cell is the treatment that patients are going to get. And we can start manufacturing RPE cells from two sources. One is the embryonic stem cell, which in fact is the one that's used today in the clinic, but we also have technology around induced pluripotent stem cells. However, the important thing is that we are manufacturing RPE at the end of the day, and we're also able to do other cells in the eye, like the corneal cells, the retinal ganglion cells, and the photoreceptor cells. Those are preclinical programs, but our goal here, as you can probably see, is we want to own the eye from front to back in terms of regenerative ophthalmology. So this is a picture of what it looks like when you have central visual acuity loss. You look at the world like this, and I can guarantee that some of us in this room are going to end up looking at the world like this if there's no treatment in the future. Dry AMD is becoming an epidemic in this country. There's about 1.8 million patients diagnosed every year with dry AMD, and eventually it progresses to the point where you can't see in your central vision. So this is important for us all. And right now there's no effective treatment option. And what we're doing, quite simply, is if you have no RPE layer, it's a single cell layer at the back of the eye, and if you do not have an RPE layer that's effectively functioning, your photoreceptors don't get any food. It's a nutritional purpose it serves. It nourishes the photoreceptors above them, and it also cleans up the back of the eye. It's kind of like a housekeeper at the back of the eye. So it keeps your eye healthy. If you lose those RPE cells, you will go blind. It's just as simple as that. So what we're doing here is addressing the root cause of macular degeneration. And this is some indication of the market opportunity we face. The Stargardt's disorder is a genetic disorder. People are born with this disease. Typically, it starts to appear in their teens, but by the time you're in your 40s, you've pretty much gone blind. And there's only about 80 to 100,000 people in the US and Europe with this disorder. In the US alone, there's probably around 32,000. This is an orphan drug indication that we're going to be targeting. We also have age-related macular degeneration, which has a number of causes. It can be genetic, but it's also environmental. This is a big market opportunity here in the United States. It's divided up into two forms, wet and dry. Now, wet is the one that most people have probably heard of because there are therapies to treat this. If you're familiar with Lucentis or Eylea as drugs, they treat wet AMD. But what we're actually going to be able to do is to catch these patients before they have the need to be treated with Lucentis or Eylea. So one could anticipate, and certainly the market research we've just finished conducting with payers and physicians, that the drug category we're entering with this therapy could potentially be as large as Eylea, if not larger. And then myopic macular degeneration is, we're in a phase 1 study going into phase 1 with that one. It uses the same RPE cells, by the way, for all three of these disorders, but the myopic macular degeneration is an environmental and genetic disorder. As I mentioned earlier, it's found mainly in Asia. So our RPE transplant therapy takes care of all three of these disorders. And we have a pipeline which is very comprehensive, actually. As I said earlier, our goal here is to own the eye. And we're going into phase 2 studies. One question you'll probably get asked is, when are we going to know how well it's working? Well, the answer is in the middle of 2016, we'll start getting data readouts from both the dry AMD trial, phase 2, which I think is probably, everyone's going to be interested in the dry AMD because it's such a big market opportunity, and we've already seen efficacy in dry AMD in the results we're just about to show you. And then the SMD trial will be completed in terms of enrollment by then. But these are long-term trials, and the SMD data readout we'll see in 2016, which Eddie will talk to, is a sort of sneak peek of the data to see how well the trial is doing. And then we have the preclinical work going on underneath in photoreceptor work, ganglion cells, and cornea. So the question is, why the eye? And this is something that is a key message too. Typically, if you're given a transplant, you need to take immunosuppressive therapy. If you're given a liver transplant, often you'll see patients on immunosuppression. This organ, the eye, is what's known as immune privileged, which means that anything you put into the eye is unlikely to be hit by the immune system of the body. That's because there's a barrier between the blood system and the eye itself. And so it's immune privileged, which means that any transplant that goes into it is less prone to rejection. Now, just as a matter of caution, we are actually running studies with immune suppression in them. The AMD trial that's going to go into phase 2 does have 3 levels. There's 0, immune suppression for 6 weeks, and immune suppression for 12 weeks. But in patients we've already treated with this transplant who are not on immune suppression today, we've seen that the transplant is still effective after 3 years. And the eye is just the right place to go. There's only one other site really that is of significance that is immune privileged, and that's the brain. And some of you may have heard the blood-brain barrier. Well, that's why it's immune privileged. The other thing which is important, it's a compact structure, which means that it doesn't require many cells to treat. We're only giving a couple of 100,000 cells, maybe 300,000 cells, to treat the back of the eye, and that means that you can give a very small dose in a small volume. So the dose of the injection that goes into the back of the eye is about 150 microliters. And one other thing which is important for those of you who are familiar with breakthrough stories in biotech or anywhere else, this delivery of this therapy, the cells are actually going to be given by a retinal injection, which is a technique that's already used by physicians today. So we're not asking them to do something they don't know how to do. We're not asking them to retrain or learn something they can't already do. It's a straightforward technique. And lastly, which is important for Eddie's point of view, is that the clinical outcome assessment tools that are going to be used here are already validated, which is important when you're looking to get a product approved. You don't want to be going out there with a clinical investigation where you're not quite sure how to evaluate the patients. In this case, we know how to do that. So the market opportunity, I won't spend too much time on this slide. The orphan drug indication Stargardt's is 32,000 patients. They're diagnosed and managed by retinal specialists, and the retinal specialist category of physician is limited to just a couple of thousand in this country, so it's very easy to target them in terms of a sales force. There are no approved therapies that stop Stargardt's, absolutely none. And the pipeline for the candidates to treat Stargardt's is very limited indeed. In fact, the nearest one to us is probably the StarGen program, which is run by Oxford Biomedica. They have a deal with Sanofi, but we're ahead of them in terms of treating patients. This is a gene therapy approach to treating Stargardt's, but fundamentally, if you don't treat the RPE layer and fix that problem, gene therapy isn't really going to be very helpful. So in actual fact, our therapy itself would be synergistic with any gene therapy approach to most eye diseases. Age-related macular degeneration. This is the big one. There's 1.8 million dry AMD patients being diagnosed every year in this country right now. And patients are getting younger and younger as they're getting diagnosed too. So it's a disease that becomes prevalent as we all get older. But not only that, but patients are being diagnosed earlier on now. And in the late form of dry AMD, you get geographic atrophy, which leads to your vision loss in the center. We estimate from the market research that we just conducted, there's probably up to about 165,000 cases of advanced dry AMD diagnosed every year where central geographic atrophy is present. And this is the target we're going after in the clinical trial program. And severe dry AMD is important, and this is easy for people to understand in terms of the value of this market, because it's the precursor to wet AMD, which is what both Lucentis and Eylea are used to treat. And the pipeline interest here is a little bit higher, obviously, than Stargardt's, because the opportunity is much larger. But probably the most significant competitor potentially would be Roche for this. But even then, the Roche therapy would be synergistic with what we're doing, because they don't fix the underlying problem of the RPE degradation. And the other thing which is important for a company like us, biotech companies or specialty pharma companies always have a tough time accessing a primary care market because of the number of sales reps you need. With this particular disorder, macular degeneration, you're looking at a very focused group of physicians, probably only 2,000 in this country. That means you can target them with a sales force that's probably in the sort of 20-person range, including MSLs. And the fact is that it's a very cost-effective sales force to manage if we want to do this ourselves. Our intent, by the way, is to certainly go to the market directly ourselves for this therapy. And the physician outreach can be achieved quite readily. In actual fact, the Lancet article that came out in October led to a lot of incoming calls to the company. And when you have a novel therapy like this, the physician outreach is relatively easy when you get a publication in a major medical journal like the Lancet. They all read it, and the data speaks for itself. The second thing is that the technology to administer the transplanted cells is already available. And like I said earlier, we're not having to retrain surgeons on how to give this injection. You only need a small dosage for the treatment. We're talking about hundreds of thousands of cells. And lastly, there is a significant unmet need for this therapy. So it's a really good opportunity to treat earlier stage AMD before patients get to the wet form. And I think I'm going to turn the presentation over now to Eddie, who'll take you through the clinical programs. He'll actually show you pictures of the eye, and you'll see how the technology is administered, and then you'll see the results of the trials that we just conducted. Thanks.
Great. Thanks, Bill. So good morning, all. It's great to see familiar faces and new ones. And just by way of background, ophthalmologist by training. It's rare that in a career span you get to work on a project that has potential impact for tremendous numbers of patients. So, I mean, if you think about 1.8 million people being diagnosed with the disease, essentially you're filling Giant Stadium every 2 weeks with people going blind. Every 2 weeks. So, in terms of the clinical development program, let's talk a little bit about the RPE. So you can see that the retina is basically a 10-cell layer thick tissue. The very bottom is a single cell layer called the pigment epithelium, and that's the tissue that we're transplanting. It has a broad range of functions, as Paul started to mention. Metabolizes vitamin A from the bloodstream as well as within the eye to allow you to see. It's required for that. About 10% of your photoreceptors, you can see the photoreceptor layer, the rods and cones, are ingested by the pigment epithelium every day. In the case of patients with Stargardt's disease, the outer segments actually are toxic to the pigment epithelium and cause it to die over the course of several decades. And our treatment would replace that function in those folks. So essentially, it's a critical function. It also secretes a number of survival and growth factors, including VEGF, and its overproduction results in the wet form of the disease. So let's talk about the business end of the eye. This is how the eye looks to an ophthalmologist in the clinic when the eye is dilated. You can see their blood vessels, the optic nerve, and then in the very center, about a 6 by 6 millimeter area called the macula. That's what you're using to see the page, and the area of highest acuity is the fovea. That's what we use every day to see, and if you lose that, vision drops immediately into the less than driving range. If you lose more than the fovea, then you're talking about legal blindness. Now, just for comparison, on the left, you see a normal fundus, and it's got a characteristic appearance, and it's got sort of a darker spot in the middle. That's the fovea. And then as you move to the right, you'll see that there's a patient with dry AMD. This is someone whose vision is less than 2200, probably 2400 or less. They have an area that you can see kind of right in the middle where the pigment epithelium's been lost. It's like a giant pothole. And our therapy essentially replaces that and allows the vision to drive on, if you will. The next picture of Stargardt's macular dystrophy, you can see that there are these sort of yellowish flecks scattered around the back, and that's because the RP is becoming overwhelmed with metabolic stuff that it just can't handle. So it just, over the course of time, you see more of that, as well as, it's hard to see here, but there's an area, again, of loss in the very middle. And finally, myopic macular degeneration is a condition where the eye is abnormally long. And as a consequence, you can see in this patient's macula, they're about 20/40 here, and that area that of yellow has enlarged so that they're legally blind within about 5 years. And this happens in when these patients are in their 40s. So devastating diseases in terms of life quality and productivity. So how does vision work? Essentially, light's bouncing off the page to the full thickness of your retina, stimulates the rods and cones. You can see that the pigment epithelium, that layer of green cells, is intimately in contact with the rods and cones. The signal goes back through the middle layer of the retina and then back to the brain. So in the setting of macular degeneration, that support function that we've been talking about is lost. You get degeneration of the photoreceptors and loss of signal transmission. Our proposition for these patients is to take a suspension of the pigment epithelial cells and inject them right under the retina. They go and find a place to sort of begin functioning, and ultimately what the thesis would be is to reestablish those connections that are necessary for vision and allow the patients to reestablish vision. So goal of our studies has been obviously to establish that this is safe. Up till now it's been technically challenging to do this, almost essentially impossible. But the first hurdle was to be able to do this, and it's been done. And the second is to say, well, what does it do to vision? Does it destroy vision? Does it enhance vision? So this is just a quick schema of the study design. We've tested several doses ranging from 50,000 cells to 200,000 cells. We're at the highest dose level right now. There's a periodic review by an independent safety monitoring board. And so far, so good in terms of safety. Most of it's been related to the use of systemic immunosuppression. Paul alluded earlier, particularly in older patients, if they've got to take transplant drugs to keep the graft, as it were, because this is a transplant. It causes problems for them. So first level of evidence is what happens when these cells are injected. And I think what you can see, if we compare the baseline photograph to the middle panel, is that there's this big area of atrophy. This is a patient with Stargardt. You can kind of see these yellowish flecks I referred to earlier. And then you have, over time, and this is obviously one image in time, an increase in the number of cells that are pigmented, the retinal pigment epithelium. And on the far right, you can see that there are these sort of two little bumps here that increase in number over time. So the cells, when they're transplanted, they establish themselves and begin to grow. Let's talk a little bit about this Lancet publication. So we studied 18 patients who had 6 months or more of data. As Paul mentioned, we've got a couple patients who are out to 3 years. The Stargardt's patients, because this is a juvenile onset disease, they're younger, but they've had profound vision loss. So you and I can see 100 letters pretty easily, or better. These patients only have 5 to 6 letters, so they're profoundly vision impaired. The dry AMD patients, they're still quite profoundly impaired, but less so. And there's some asymmetry in their disease, whereas because this is an autosomal recessive disease, you get the disease impacting both eyes in a fairly symmetric fashion. No gender or racial predilection, but that's how it broke out. And let's talk about how they did with respect to vision. So The anticipation was that we obviously wouldn't have patients experiencing loss of vision, but in fact what was seen was patients gained vision in both diseases, and it was stable over time. Pretty amazing. So the yellow line is the treated eye, the blue line the untreated eye, and the green line the difference between the two, and what you can see in terms of the difference between the vision, and this is both the case for Stargardt's, though not to the same magnitude as dry AMD, is that it increases over time. It comes up pretty early and then remains stable for the ensuing 12 months of observation for most of these patients. And as I mentioned, there are patients who are out as far as 3 years who've sustained those improvements in vision as well as that pigmentation. Now, we talk about 13 patients. We excluded 5 patients who developed cataract, but if we put those back in, the difference is sustained, but obviously the magnitude is not as great, and we'll accommodate for that in the course of our development program. So in terms of leveraging what we've learned in phase 2, as Paul mentioned, we're leading with the Stargardt's program. The trial is designed as a 100-patient trial, and it's got 2 primary endpoints. It's a co-primary, so we'll look at the change in vision because that's what the patients care about particularly. But we'll also look at this atrophy, because atrophy is equal to loss of vision, at least in the mind of FDA and the European regulators. That's an open label, and we're going to use the fellow eye as the control with a little bit of dose escalation. The enrollment timeline is on the order of about 18 months. For the AMD trial, as I mentioned, the older patients tend to have problems with the immunosuppression. So as Paul mentioned earlier, and as I'll reinforce, the thrust of that study is to optimize the immunosuppression. And we'll also look at efficacy outcome parameters, obviously, and it'll be pretty exciting. Finally, the myopic macular degeneration trial, epidemic disease in the Asian countries, will be a single-center trial at UCLA. This is just a brief schema to mention the fact this is a commonly performed procedure about 300,000 times a year. That's Dr. Schwartz at the operating microscope and the equipment that he uses, and the picture of the eye that's received the injection with this cannula that I talked about earlier. We have, as Paul mentioned, a range of programs that go from the front to the back of the eye, and obviously the dream for this program that we're conducting right now is that it essentially becomes as common as cataract surgery is for the front of the eye. Because cataracts can be easily cured, but obviously A and B cannot at this point. So I'll turn over to Ted.
Thanks, Eddie. So a couple quick words on a few topics here. One of the key items I want to speak about is manufacturing. A lot of small companies with great technology can trip on supply, and this is an area where Ocata's made a very significant investment. Not only does it give us the ability to really control our destiny as we fund these clinical trials, it's also given us a lot of IP and a lot of protection from competition down the road. Being able to differentiate or really own these master cell banks, which we have in our facility in Marlborough and remote locations, and knowing how to go in, we've got the people that know how to go in, take these cells, induce them to proliferate into expansions of multiples of what they currently are. And then the real special sauce is being able to tease them into differentiating into exactly what type of cell we want. A stem cell can be any type of cell in the body. ACT or Ocata's scientists and the manufacturing folks know how to force that cell into what we ultimately want it to be. In this case, in the case of all these programs, we're speaking about the retinal pigment epithelial cells. So once we go into the master cell bank, take some supply of embryonic stem cells, let them expand, we induce them to change into an RPE cell. Then we've developed a lot of testing. As you can imagine, the FDA, before we were allowed to go into human subjects, wanted to make sure that, and the company wanted to make sure that these cells actually were terminally differentiated RPE, that they weren't going to turn into something else. And so the company, being the first, had to develop all the assays and all the testing that would be required to confirm that. The FDA has gone through, is comfortable, and as Eddie mentioned earlier, we've treated 38 patients safely. Again, safety, good IP, good protection, a lot of know-how that the rest of the market would have to try to catch up and find. Ultimately, once we confirm that they are RPE cells, we have methods to really support their further expansion so that we can turn them into inventory for the treatment. This process oversimplifies but sort of lays out what's currently about a 4 to 5 month process at the company and yields 1,000 to 1,200 doses of the therapy. All the trials that Eddie mentioned that we're going to be embarking on and supporting over the next 2 years will require about 200 doses. So, as I mentioned earlier, this is a small company, but it controls its destiny. We've got the manufacturing piece down. We feel that we've got a lot of the foundation, the fundamentals of the technology very well mastered, and all we have to do now is do the work to show that we can make enough cells and enough doses to serve the entire market. As I mentioned, one of the nice byproducts of all this effort has been a lot of IP. So this is just one schematic. We could do a whole seminar on the company's IP because we have really been working hard in the space and developing a lot of technology throughout the company's 15-year history. The quick overview here is from the initial derivation of the human embryonic stem cells through manufacturing. I mentioned product release assays, pharmaceutical preps, and then all the way into the eyeball. So we say from the embryo to the eyeball, we have a very significant IP estates that each step of the way has not only strengthened our position, but also extended the life of the technology and what we believe will be the life of the leadership of the company. We are looking to raise approximately $60 million that'll fund these trials, 40 of which will go into the clinical programs Eddie mentioned and the manufacturing of that supply. The balance is split about 2/3, 1/3 between G&A and basic research to advance the preclinical programs. Currently funded with an equity line from Lincoln Park. It's not an ideal long-term financing, but it's been a good bridge to get us to here, and it's clean. The balance sheet is clean. The cap table's clean. No preferred, no debt. As I mentioned, we're active dialogue with NASDAQ. So there's a lot to be done. Q4, this is when we capitalize the company and uplist the stock to NASDAQ, and also when we're going to treat our first SMD phase 2 patient. And then we go into 15 and 16, the real work begins. 15 is really a building year. We're going to be expanding from 4 clinical sites in the U.S. and 2 in Europe to about 20 and 10 to get all these patients treated. So the first 6 months, 8 months of 2015 will be building those, and then the patient flow will really begin in earnest as these sites are up and running and patients come through and receive the treatment. Paul mentioned earlier critical milestones in 2016. You look at Q2, phase 1 of the MMD program, a readout on those patients. Half of them will be treated, have 6 months. They'll be treated, and then 6 months of follow-up. Q3, very big period of time for the company, SMD and AMD. We're pretty confident that once we've treated 50 patients and seen them with 6 months of follow-up, we'll have a very, very good idea of how this therapy is working. Thank you.
So that's the Ocata story. This is a reinvention of a company. I think that there is a lot of hard work that we've got to do in the future. But I'm really excited about this. I left a company that was pretty successful to come to this one because I'd seen the data that was out there in the Lancet publication. In fact, all 3 of us came to this company. So you've got a new management team effectively running this business, and the data speaks for itself. I mean, I saw that. I joined and signed up. We just need some investors to sign up now, which is your guys' job to go and find those for us. We're about to initiate phase 2 studies. One of those is potential registration study, certainly for Europe, in Stargardt's. Dry AMD, that's important because if we can repeat the success of Lucentis or Eylea, we're going to do really well. We've shown for the first time in any disease indication that cell therapy can be used in patients, and certainly in the back of the eye, and the data in the Lancet support that. We've got, as Ted mentioned, a really strong, in fact it describes it in here, a robust intellectual property position, and that's because the company is one of the originators of this type of technology. And lastly, we're going to be uplisting to NASDAQ when this transaction's done. So hopefully we're going to see a value uptick just on the basis of that uplist alone. But it's a really exciting time to be involved with Ocata. I'd like to thank Kevin and his team for getting us here, and look forward to getting out on the road over the next 10 days and to pricing this one towards the end of next week.